Well, good morning, everybody, and to this talk about the new Agarose-based chromatography platform. I'm happy to see that a few people made it here, even though we've heard about bus and train strikes. And actually, our second talker, Alessandra Basso, is still sitting on a train, I think. So hopefully she'll make it in time. Otherwise, we have plenty of time to spend on Agarose. So this presentation is divided into three different parts. First, I will talk about a little bit about the background of why Purolite chose to develop a platform based on Agarose. Then I will share some results of the first resins that we are producing. And thirdly, I will talk a little bit about the first range of product that we will put in the market within a month or so. So, what are the ideal resin requirements for large-scale protein purification? Well, of course, you need high capacity. That means a large accessible surface area. You want a low column back pressure, so you can run them in existing facilities. That basically means you want large beads or rigid beads. But on the other hand, you want a rapid mass transfer to get a good separation, so you need small beads with large pores for proteins. You also want to have a very hydrophilic base matrix, kind of protein-friendly with minimal or no unspecific binding to your target molecule. You also want it to be chemically stable, so you can clean, sanitize, and reuse the resins over many cycles. In addition, you want it to be physically stable, to be easy to handle and to pack in large columns. Nowadays, commonly automated packing columns, where they ideally would be able to handle pumping, etc. And finally, you want them to be easy to modify to create different types of chromatography resins. So we think Agarose is the ideal base matrix for protein purification, because it's extremely hydrophilic. It's very easy to conjugate with ligands to make different types of chromatography media, everything from protein A resins to ion exchange resins and hydrophobic interaction chromatography resins. It's also, and that's very important in industrial purification, it's very stable under alkaline conditions. You can use 1 molar sodium hydroxide, which is the golden standard for both cleaning and sanitization in the biotech industry. And in large-scale manufacturing, this is actually instrumental to quality and production economy. So if you would like to make your own Agarose beads rather than buying them, here's a crash course. Basically, you go to the beach, you collect some red seaweed, and you extract the agarose. You have to remove the agaropectin that is charged. Then you heat the agarose, and you add it to a solvent in a stirred reactor. And the shear force of the impeller, or rather the impeller speed, will define the particle size. Then you let it cool, and you will have a very soft resin. If any one of you have used agar plates, that's the type of material you will have. So you need to cross-link it to make the beads much stronger. And once you've done that, you could add functional groups to create the type of resin you want. And finally, of course, you want to pack the beads in a column for evaluation. So we've been working with this for some time now. And I just want to highlight some important design parameters. Particle size, of course, is very important from both the mass transfer efficiency perspective and also pressure flow properties. The agarose concentration, what determines the porosity of the final product. So that's also important. Less agarose, larger pores, but also worse pressure flow properties. And finally, cross-linking is also a critical parameter which affects, of course, pressure flow, but could also affect surface properties and porosity. So to cover most possible protein applications, you can't really do that with one particle size. You need 3 particle size, and this is just the common layout. People tend to use larger particles for the capture step directly after the fermenter or the cell culture reactor and clarification. And then as you get into purer products, you do intermediate purification and polishing, maybe just to remove product heterogeneities, and you would like to have smaller particle size. And our take is that the upper limit for a useful agarose resin is in the area of 80 to 100 micron because of diffusion limitation. If you make larger beads, you still have to run them at a very low flow rate to allow for the protein to both diffuse in and out of the beads. And the lower limit is roughly around 40 micrometers if you want to have a decent productivity. So with that, I should also comment on the pressure flow. What is really the need? Because there is a balance. You can make very rigid agarose beads, but that will come at the cost of porosity and surface properties. And if we look at the industry today, and all the big biotech companies, they have a fairly large installed capacity, and those plants are always rated to around, they don't use pressure above 2 to 3 bars. So we think there's really no need to make a resin that could take more than 4 bars pressure drop over the resin or over the column. And in reality, you rarely work under conditions where you have more than 2 bars pressure drop over the column. So based on this background, we have made a range of agarose resins, and I like to share some results on the 3 critical performance parameters: pressure flow properties, dynamic binding capacity, and selectivity. Starting with pressure flow, one way, or the most common way to use, is to simply plot the linear flow velocity over through the column against back pressure, and you, by stepwise increasing the pressure, you get this type of curve. If it was a completely incompressible resin, you would have a straight line. And here we've done a comparison between a GE resin, Sepharos 6 fast flow, and I think if you are in the business, GE is basically the only one of the few or the larger suppliers of agarose resins on the market. This is the GE resin, and you can see already at 1 bar it starts to compress rapidly, while with the corresponding Praesto 90 resin, which has the same particle size, so I could say Praesto will be the name of our product line. At 3 bars, you have more than twice the flow rate as compared to the GE resins. And if we look at the smaller particles, we have one Praesto Pure 45, so a 45 micron agarose speed, and compare that to Capto Impress. This particular one has 41 micrometers, and we also have an older, even smaller particle, 34 micron Sepharos high performance. You can see doing the same experiment, they more or less overlay all the way up to 4 bars. So we are very happy with the pressure flow performance and the rigidity of the beads. And the next performance parameter of big importance is, of course, dynamic binding capacity. So we've tested that with both polyclonal higG and also monoclonal antibodies using our 45 micron bead, Praesto SP45, and compared it to, on the 2 first ones here, this is dynamic binding capacity of human polyclonal higG, that is higG from plasma, compared with the Capto Impact. And we can see that while they had somewhere between close to 70 milligrams per milliliter, we reach well above 100 milligrams per milliliter under exactly the same conditions. So almost a 50% higher dynamic binding capacity. If you look at antibodies, we have some work done by Anurag Rathore, who used to work for Amgen, but now is a professor in India. And he did this evaluation for us, where he compared, or actually measured the dynamic capacity of one Mab, Mab A, had a very good capacity, almost as high as human polyclonal IgG. And then the second Mab that was somewhat bulkier, so having a larger apparent molecular weight, maybe higher glycosylation or so, slightly lower or a lower capacity, 80 milligrams per milliliter, but by increasing the residence time or decreasing the flow rate, you can compensate for that and still get a fairly good capacity at 6 minutes residence time. So we were very happy with those results. But you also have, I mean, capacity is not enough. I should say that monoclonal antibody purification is probably the largest application for cation exchangers to remove, primarily to remove aggregates, but also to decrease the amount of host cell proteins and other contaminants. So we tested that, or rather Anurag tested that in India with one antibody. Here we load about 50% of the dynamic binding capacity. And you can see this is a loading phase. So it's a monoclonal antibody that has already been purified by protein A. And then you want to use the cation exchange step to remove trait contaminants, primarily dimers or high molecular weight aggregates. And you can see that you get separation of them here. And this is what you would expect from a process chromatogram. This is what they look like. The results shows in the next slide. Yeah, so we have plotted. First, it's probably hard to see, but this starting material had an aggregate content of 4.5% aggregates, dimers, and larger molecular weight. And typically in the industry, you want to get below 1%, or ideally below 0.5% of aggregates in the final formulation. And if we plot the cumulative yield against here, recovery of protein, against high molecular weight percent of the total, we see that we get a recovery of almost 90% of the monomer below 1% aggregate, which kind of fulfills the specification, and it's a good separation. To further look at the selectivity, we did a model system, a very simple model. We simply mixed human monoclonal IgG with lactoferrin and tested the separation using a linear salt gradient. So here we compare 90 micron from Agarose fast flow with the Praesto SP90, as well the Praesto SP45, the 45 micron with the Capto Impact, and also Capto S. It's another Agarose resin that is dextran grafted, a 90 micron bead. And what it shows is that we have, for the non-grafted resins, all the resins except Capto S, we have a very similar selectivity. This is hard to see for you, but these are the 90 or the 45 microns resins, and basically they are like an overlay, identical with respect to selectivity. You see a little shift in retention, but that's all. Otherwise, same peak height, same selectivity or resolution. The one showing a different resolution is a dextran grafted resin that has a different behavior because of the dextran grafting and much poorer resolution. And if we increase the load, this was 25 milligrams human IgG, and if we increase it to 80 milligrams of IgG and still 5 milligrams of lactoferrin per milliliter resin, we see the same pattern, almost identical selectivity, except for the dextran grafted, where you start to actually lose the resolution completely from the lactoferrin. We took the 2-1. You can see here, you start to see a breakthrough of all the resins except the dextran grafted and our 45 micron resin. So we took 2 resins and loaded even more, 100 milligrams per milliliter. And you can see a clear breakthrough on the Capto Impress, and on the Praesto SP45, we start to see a minimal breakthrough after 100 milligrams per milliliter. So significantly higher dynamic binding capacity. We haven't done any studies on functional lifetime yet, but as I mentioned, reuse is really instrumental to good process economy, because these resins, the cost of these resins makes it prohibitive to use them in most cases as single-use resins. You want to use them for a number of cycles. And agarose, because of the hydrophilicity and the sodium hydroxide resistance, is actually a resin that doesn't easily be fouled, and when it's fouled, if it's fouled, it can still be very easily cleaned with sodium hydroxide. And it's been used in the industry for more than 30 years. And there are several papers published where they have used agarose resins for 300, 400, 500 cycles in one single process. And in addition, the elastic properties of cross-linked agarose beads make them very durable and easy to pack and repack in modern intelligent chromatography columns. So with that, I move over to like a little direct comparison of the product that we will put on the market within a month. So if we compare our 90 micron resin under-rivetized, so no ligands, with the Zephyrus 4 and 6 fast flow, we have much better pressure flow properties, and the porosity, the pore size, will be somewhere between Zephyrus 4 fast flow and 6 fast flow. For the smaller particle sizes, 65 and 45, there is really no Zephyrus equivalent, because GE has decided, at least currently, they don't sell the high-flow agarose under-rivetized. They only use it for internal to make other products. We have the ion exchange to the 90 micron. We have much better pressure flow properties and slightly higher capacity for albumins and larger proteins. Once again, with the 65 is just a resin in between the larger and the smaller. There's really no GE equivalent today. And finally, on the 45 micron Q and S, cation and cation exchangers, we have equal pressure flow properties, but much higher dynamic binding capacity. For antibodies, typically in the range of 50% higher dynamic binding capacity. So what we will start with on the market is the 3Q, the strong anion exchangers, 45, 65, and 90 micron. And we have some specifications here. Pretty high binding capacity. The binding capacity, of course, the dynamic binding capacity will be a function of the flow rate and the the flow rate, the residence time. So these are just kind of estimated values for a moderate flow. And the same for Praesto SP, very good values for higG. And we have the ionic capacity, which is in the similar range as on most ion exchangers, agarose based on the market. I also mentioned that we will sell these high flow agarose resins directly on the market. One application is gel filtration of macromolecules. So for example, if you work with virus or virus-like particle purification, it's common to have a gel filtration step where the virus will pass through the resin in the void, and you will get proteins delayed in the resin. The other application is to make custom designed resins, because then we have a very good base matrix, and from that we can make all types of different resins, even custom designed resins. These resins will be manufactured in the UK, and we've already built a pilot plant, and we typically run all our plants and labs under ISO 9001. So this pilot plant will be used to produce the first range of products, but we have already started to design a full-scale GMP-like manufacturing plant, also in Wales, that we plan to have online by the end of next year. So within the next months, we will put these 9 products on the market. We will start to supply them in lab packs up to half a liter and larger amounts on request. We will also sell them pre-packed. We work together with Atoll, a German company that custom packs resins for most resin manufacturers today. And we will start with 2 formats typically used for process development and early testing of resins, as well as a robo-column that is used for high throughput process development with most commonly Tecan robots. But this is just the start. This is what we will put on the market a year. Next year we will expand this portfolio to cover protein A affinity chromatography, hydrophobic interaction chromatography, and also a concept where we can produce custom designed resins in our pilot lab. In parallel, I haven't talked about that today, but we work these products we do in a classical way, like most resins are produced today, in a batch reactor, stirred reactor. In parallel, we are working with the jetting technology. So Purolite makes polymer resins already today in large scale, in really large scale, using jetting, and we are applying this technology for our Agarose material as well. And the advantage would be that you get a more narrow particle distribution, which translates to lower production costs for Purolite, but also better pressure flow properties for the users. So that was the end of this presentation. We have, as I said, our next speaker hasn't arrived yet. We have plenty of time for questions, if you have any questions.